I'd like to apologize in advance. I'm worried about my internet connection tonight. It's been kind of up and down today, so it could at some point go and be done.
While other people are talking now. Okay. Ready for
Ready.
the big show? Disclaimer, disclaimer, disclaimer. The following hour of programming requires listeners to sign a waiver for any injury incurred to your mental state in the course of listening to the following hour of programming. If you are still listening, it is assumed that you understand the gravity of the situation you have gotten yourself into, and there will be no further warnings. The dangers ahead on This Week in Science, coming up next. Cue the music.
Or
Music's not happening, so we'll have to deal with
Okay.
that later.
I will cut it out. I will put it in
I hear
myself.
crickets.
Just thought I'd try something. I thought I'd try something new. All right. End of music. Go. Hey, everybody.
Oh, yeah,
It's
it's the
time
part
for
where
This
we
Week in
sing hey,
Science. It is time once again for This Week in Science. I'm Dr. Kiki, and I'm here with Justin and Allie, our intern. Say hi, Allie.
Hi.
she's now a co-host, isn't she?
I guess this would
You
officially
can't really
make her.
be an on-air intern.
Why not? There are no rules except for the rules that we make and then break. So, welcome,
Yep.
Allie, to the online video show. You're no longer the person quietly just writing the show notes. You're here. Yay.
Yay.
Yay. So excited. So, on today's show, we have all sorts of science news as normal. We've got, what do we have today? I have stories about a twin mystery, about solar siblings, and bird brains. What do you have, Justin?
I have why eating aphids is as good as eating tomatoes. Why children, just interesting notes by psychologists who are watching children. And a couple bacteria stories. One's a fungus fighting a bacteria, the other's a really groovy new bacteria that they're focusing on to save us from ourselves.
Awesome. And Allie, what stories did you bring today?
I've got extra empathetic men and gardens on Mars.
Very nice. I'm looking forward to those stories.
Mhm.
I'm always looking for that extra empathetic man in my life. But to get it all started, let's talk about twins and a mystery. It's like, what is it, the Hardy Boys? Is it Nancy Drew, the Hardy Boys?
Huh?
The Hardy Boys. Were they twins? Do you guys remember?
I don't think they
Anyway.
were twins, because wasn't one of them an older brother?
Oh, maybe.
But I guess twins have older brothers too, or
Yeah,
sisters.
my childhood literature is passing me by. I don't remember anymore. But anyway, in the most detailed genetic investigation ever of multiple sclerosis, researchers have only found more questions. Isn't that wonderful? So, researchers publishing in Nature magazine this last week, geneticist Stephen Kingsmore says, in terms of the cause of multiple sclerosis from genetics, quote, we find no smoking gun on the genetic level. And the research itself is pretty interesting. The fact that it's the most complete study, I think, is the really, really interesting part. They took, I think it was 3 groups of twins. They had one set of twins whose genomes they analyzed completely. It cost $1.5 million, and it took 18 months to sequence the 2.8 billion DNA units in each of the twins. And in addition to doing that, they also did genetic analysis to determine whether each of those DNA units came from the mother or the father. So they're actually determining the parental inheritance to figure out where they came from and what they would have gotten them from. One twin in the pair had multiple sclerosis. The other twin did not. Multiple sclerosis historically, research has shown, just based on a probability level, that there's high likelihood of a genetic cause. If you're a twin and your twin has multiple sclerosis, the the likelihood that you're going to get it goes up by 25%. And if you have a family member with multiple sclerosis, I believe your chances increase by like 6 times or something like that. So genetics is, there should be something there, but they've done this analysis, looked at all of these genes, they looked at the parental inheritance, and then they went even further. They had 3 more sets of twins who they analyzed specific genetic factors in their white blood cells. So they looked at where was it? They looked at chemical instructions, the epigenomes. These are the instructions that are in the DNA that turn genes on or turn them off and allow them to be transcribed into proteins or allow them to be activated, and transcriptomes. And transcriptomes are the genes that are being transcribed or that are actively being coded. So they took the genes and then they took the instructions that turn the genes on and off, and then they actually looked at the chemical record of which ones were being turned on and off, and they found nothing. No, they found no genetic smoking gun. They found, oh yeah, okay, they have some common genes in here, but they have nothing genetic. And so the researchers say this was a technical tour de force and potentially represents a new way of looking at disease states. However, there must be a non-genetic factor, probably environmental, that combines with the known genetic and environmental risks. And they're looking at the fact that this was just one of the most advanced studies of our day, and they have a mystery on their hands. So they're
Wow.
just, yeah, it's just amazing, right?
That's wild.
Yeah, I think it's a really cool study. Multiple sclerosis is a disease that it it attacks people's the immune system attacks yourself, your nervous cells, and causes all sorts of debilitating things to take place. And if we could find a cause, it would just be so. I mean, it would help so many people. That there's got to be some trigger somewhere, and the fact that one twin has MS and the other doesn't, and they did the entire genome analysis to find out, yes, they are identical twins. The genes are identical pretty much. Why does one have this disease and the other not? And there's got to be, what the researchers say is that one had to have been exposed to the perfect combination of environmental triggers. And the researchers go on to say that maybe it's the fact that they only sequenced one set of twins. And then they looked at this smaller subset of 3 other sets of twins. And maybe if they looked at a dozen or 2 dozen or 3 dozen sets of twins, that maybe they'd find something, but that's not where this study is right now. So we just have to wait for sequencing costs to come down, maybe. I don't know. What do you think, Allie?
Interesting. Did they do any sort of tests on the environmental conditions or anything yet? Or did they say they were planning on any that you know of?
That's something they haven't looked at. They've only looked at the just the genome. So there could definitely be some environmental factors, but you have to find a way to go back in time, because it doesn't necessarily mean that the trigger is still there in the environment, but that it was there at one point in time and started this immune storm.
Yeah,
Where
that's
they
like,
started.
my goodness, that could be just such an impossible task to try to figure out the environmental factor after the fact on something that is,
Right.
yeah, something is thought to be, I mean, that is thought to be have genetic causes, but then environmental triggers to that have to turn the genes on in just the right way. And where was the other twin that day the gamma ray came down and like it's actually pretty awful, because it makes tracking it means that it's going to be much harder. At least at this point, it looks like it'll be much harder to figure out the root causes.
Yeah, the finding the causes is going to be immensely hard. I mean, the sample size is something that I think is really important here, and I think the researchers are admitting that they probably just haven't sampled enough people. Even though you're looking at 2.8 billion DNA base pairs, you're still only looking at two people, and one of them has it and one of them doesn't. So if you could increase that, repeat it over and over and over again, that might actually make the difference. Um, yeah, I don't know. I think it's a huge question. Why does the body just start attacking itself? Why does the immune system go crazy all of a sudden? We don't know. We don't know. What you got, Justin?
I've got to go sneeze.
Is that
Allie,
what
have you
you-
got a story handy?
Oh yeah, sure. I got a.
It's
I
good
have a
timing,
story
right? Because you're going to be
about.
talking about. Sorry about that. I was going to say good timing for your story, Allie. Actually.
Oh yeah. I have a story about empathetic men, or more empathetic men, actually. A new study has found that the neuropeptide oxytocin enhances male empathy. And this is the chemical that increases the feelings of like love and trust between people. It's also released during orgasm. It also causes labor pains and the emotional bond between mother and child. But a new study has found that if they basically spray it into men's nostrils, they become more empathetic for a while, similar to how women are all times. They found that
Wait, women are at all times. Hey.
yeah. That's scientifically proven here. They found in a test of 48 healthy males, half of which received oxytocin nose spray, and half received a placebo. And the half that received the spray, and then they showed them all pictures of kind of emotionally charged situations, like a crying child or a girl hugging her cat, or a grieving man. And then they were told to express the depth of feeling they experienced for the people shown. And in their results, they found that the men that had been sprayed with oxytocin had more stronger reaction, had a stronger reaction to the different pictures, and they also tested women, and they found that they were a similar level to the men with oxytocin. Yeah, so basically they found
Empathy
that
in a bottle.
I know.
I'm sure a lot of women would want that, want to have that to distribute to their significant other.
I think that brings up a question, though. I mean, are people going to be running around with little spray bottles of oxytocin, spraying them up people's
know.
noses?
I don't
Feel what I
I don't know
feel.
I don't know how long it lasts either. The study didn't say, but that would be interesting too, to see how long it lasted, to test them the next day or whatever.
Right.
But yeah, they also did a second study where they looked at, basically they had people do a simple test, an observation test, it says, and correct answers, they got an approving face on the screen, and wrong ones, they got a disapproving face. And basically people, or the men who had oxytocin, learned faster, or they were more receptive to that training than the placebo group, which I guess basically is showing that they were more impacted by seeing negative faces or positive faces, so facial feedback. Yeah, and basically, this is from a study in Cambridge Barham Institute in England. It says, once again, the oxytocin group responded clearly better to the feedback in the form of facial expression than did the placebo group. So it's a similar study, basically, that implies the same sort of thing. This one, it's called social. It's more of a social interaction sort of thing, but same idea. And they're saying that this might mean that the hormone might be useful as a medication, actually, for diseases such as schizophrenia, which is involved with like social withdrawal or social approachability. So yeah.
Yeah, so people may be looking at with schizophrenia or other disorders where they see faces as maybe more disapproving than they actually are.
Okay,
This could
yeah.
potentially alleviate those feelings, maybe make
Hmm.
them a little, more likely to find faces as approving or, not as negative as maybe their brain is making them out to be. I don't know. It's an interesting idea. Yeah, I love this. What is the in the story about here talking about the amygdala and how the amygdala is involved in this, and that there might be that does it seem as though the oxytocin is feeding back and affecting the amygdala itself, your fear center?
Yeah, basically, they also found that they were using female patients as well, and they had two of them in their study who were suffering from a defect in the amygdala that you're talking about. And they said both women reacted markedly worse to approving or disapproving faces in the observation test than did the other women in a control group. Um, so yeah, it seems that that is what is being used or playing an important role. Um, and they're saying that the emotional empathy was also affected in these women. So it would make sense that the oxytocin interacts somehow with the amygdala if it reacts with empathy. So yeah, it would make sense. It doesn't say here exactly, but yeah, it's implied.
Yeah, I still think it's kind of I love finding out about how these hormones work and how a very simple substance, one compound, oxytocin, can have all of these behavioral effects, and that it can be so dramatic. And that at the same, if we learn how to use it, and if it becomes something that people want to use, it can be a love drug, or what it, I don't know.
Don't we have
I'm
enough
starting to
of
think about
those
the
at
drug
this
of
point?
abuse.
Don't we have enough love drugs at this point that we probably don't need any more? I don't know. I think one of the problems is when you say it like that, though, is that is what happens. I mean, we've had a lot of drugs that had very powerful, profound, effective therapeutic benefits become illegal because they became recreational, which is really odd to me. I can see how a therapeutic drug can become banned from recreational use. I still don't
Mhm.
quite understand how something that starts out therapeutic is banned for therapeutic use because it becomes a recreational drug. But that happened to LSD and the MDA and the ecstasy
Right.
as
Well,
well.
it's at the point that anything becomes a drug of abuse, or goes beyond, or it's, yeah, black market kind of stuff. We want to that this is socio political
Is that what it
commentary.
is? It's for the political reasons?
It's political reasons. It's all money. It all comes
Yeah.
down to money and control. That's all I
Yeah.
think. I don't know. I don't know. Yeah, I mean, if it's a therapy, if it really were just a therapeutic drug and it were being used that way, and people use it recreationally, and it doesn't go too far, then it's not. I don't know. I know. I think the problem is when drugs start being seen as hurting people and no longer having their therapeutic use. But at the same time, right now there are lots of drugs like LSD and psilocybin and marijuana and ecstasy, MDMA, and ketamine that are being looked at for being returned to therapeutic use. There are studies ongoing for like PTSD and other emotional issues, and um, yeah, a bunch of stuff for psychological therapy and therapeutic uses that they were made into Schedule 4 illegal substances. But now let's see if we can actually bring them back and show that what psychiatrists were saying 20, 30 years ago about them being beneficial. Maybe we should bring them back and actually let's have some scientific studies that show that they're actually useful.
I
Yeah, and
think
I
it's
think
really
Go
interesting
ahead.
too the naturally occurring chemicals like brain chemicals now that they're discovering them more and more that they could be distilled and yeah like manufactured and distributed basically and that people could manufacture emotions that they would want to have maybe like they could
Exactly.
buy the drug that could cause that emotion and it's not even a drug that causes it a foreign type of drug that you're putting in your body that affects it but it's the actual chemical that's made in your body. I think that's really crazy that could happen more and more as more are discovered and stuff.
Yeah, well, that's what consumerism's for. You buy something
Yeah.
that gives you a feeling.
Yeah.
I think if you want to solve crime, and you can pick any crime, it doesn't matter. It's just crime in general. I think anybody who goes to jail, you should give them LSD while in prison. I think you wouldn't. I think that would leave quite an impression on anybody in prison, so where they would never get themselves in that situation again. It would just be like a mandatory thing. Like, okay, you're in jail. You're going to be here for a little while. While you're thinking about that, here's your LSD, and we'll put you in a room and let you think about it overnight. And I think most people would come to the conclusion that they would really want to avoid that. And I don't think it would be torture. I think it would just be a whole lot of perspective. They used to do that, though. They actually did that. They actually did that study back in the 60s.
Yeah, but it was one of those things.
And what do you call it? Revitism? The thing where they don't, where they would normally do it again. They didn't do it again. They had low return.
Recidivism rate? You got it.
Revisiting isms. They're just words. We know what they mean. We don't have to articulate them.
Yeah. With this idea of like the molecules and drugs that can be used, I mean, this is what you eat food. I mean, this is the point you made, Justin. I mean, whatever you eat, whatever you take, I mean, there's a lot of self-medicating just in terms of drinking caffeinated beverages, smoking cigarettes, eating fatty foods, whatever it happens to be. But if we can actually start creating specific molecules like oxytocin or, any number of hormones or neurotransmitters and just make them in a way that can be delivered to the place in the body where they need to go. I mean, are you going to be thinking faster? Are you going to be smarter? Are you going to be nicer? Are you going to be more aggressive?
But it's that need. That's the need is the whole question. The need is that really tricky part. Like, what's the need? Is the need because you need to work longer? So you need something to make you more attentive or keep you from, keep you awake longer or be able to focus on one thing at a time better? I mean, is that the need? Is it based on your job? Is it based on what's required of you? And then if you're the only one not doing those drugs or having those therapeutic deliveries, and then you're not competing as well, maybe the problem is you need to go see your doctor. And then your doctor says, oh, well, here's the problem. You're not on any of the juice.
That's right. That's right. You're going to be at a disadvantage at some point in time because you're not caffeinated or you're not whatever it happens to be. Yeah.
You're the one baseball player that didn't take steroids and you didn't make the team. Maybe that's because you didn't take steroids and everybody else did. I don't know.
Let's go to another story. You have one, right, Justin?
You would think I did, having said that there were, but maybe not. Okay, this is the aphid thing.
And if you just tuned in, you're listening to This Week in Science.
And it is time to go to the break.
No, it's not. I was just saying, just redo your story.
Oh, yeah. Okay, so I didn't realize this. This is something new. One of those had no idea this was even possible. But the aphids are making the carotenoids.
Carotenoids?
Carotenoids. That's the stuff in tomatoes. That's why you're supposed to eat lots of tomatoes. Because they'll help prevent cancer because they've got those red, yummy carotenoids in them. It's part of the thing you can tell, right? The carotenoids. They can make them. That's pretty cool. So this is
end of story.
They're the only. Yeah, that's the only life form. Every other form of life. Every other piece of biology that makes these are a plant. This is completely thought to be something that's created in plants. One life form that we know of in the animal insect kingdom that produces this. It's the aphid. It's usually considered a pretty horrible thing to have in your garden. But now next time you find aphids, you can say, that's pretty unique. That's a pretty rad genetic trick to be able to, because this is something that's an essential requirement. We have to eat some certain amount of plants to get that into our system, because we can't produce it, and they are what do you call it? They're an essential nutrient.
So there's somebody in the chat room who's asking, Bird to Brain is asking, would aphids make a nice sauce on pasta then?
We should try.
I know.
should try.
Do we want to eat them?
Tomato. Well, I don't know. I've never tried an aphid that I know of. Tomato worms. You know what they taste like?
No.
Tomatoes.
Really?
I would assume aphids would taste like perhaps roses, or maybe it would just depend on where you found.
Probably.
What does that sound? Okay, so there was that story, which nobody's impressed with that one. Okay, so the other one is that we're working with purple bacteria. These are single-celled microscopic organisms that are usually in aquatic environments, bottom of lakes, coral areas under the sea. They're found now to be the most efficient converter of solar energy that science can find up to this point. Much more efficient than any technology that we've worked out. Much more efficient than any other algae or plant. So they're gonna begin some research trying to convert this into maybe even something that can produce energy for us.
Yeah, I love the idea of bio creating energy. So how do we look for the most efficient organisms? Can we get them to do work for us, or can we figure out how they work and be able to mimic that process chemically? Or
it's a single-celled creature. I would think it wouldn't be too difficult for us to tear that one apart and back engineer it, but
yeah, but single cell in terms of that's highly complex when it really comes down to it. In terms of energy photosynthesis, we're looking at electron transport. Sun comes in, boom, hits stuff. You have electrons bouncing around following a quantum path, and then you have oxygen pop out the other end. There's a lot of interesting, very technical stuff that's going on in there that maybe we can get close chemically, but until we really figure out what's going on, we're not going to reach the same efficiencies as Mother Nature.
Right. Although I do like the idea of using bacteria and algae much, much more than using corn or rice or anything that we would eat. And I think that just puts us all in a bad spot.
I'm gonna go ahead.
Now, if we have to compete against cars, robots for food, I think all of us will lose.
You think so? I don't know about that.
Yeah, I think we'd all go down.
More
sushi restaurants.
Yeah, they've already shown that with the studies of corn efficiencies in biofuels, that it's not efficient. That the too much land, too much water is required, transporting stuff, it just doesn't work. And then you have to share that versus the actual food crops. There's only a limited amount of space.
Yep. And if the cornflakes if the cornflakes aren't as valuable as the gallon of gas, then you wouldn't have the cornflakes on the shelf. They would probably have to match. They'd have to get up to some point where they were equal. That what you ate in corn was worth its weight in gasoline. Basically, it would have to end up at that point eventually. Whew.
Whoosh, whoosh. And I think it's about time to go to the break, people. I think we've done about the first half of This Week in Science for this week episode. What are we, 265 now? This is kind of crazy. Anyway, we'll be back in just a moment. Stay tuned. Oh, we have...
I built something that'll change the world. I'll win the Nobel Prize and maybe even a girl. It doesn't need a battery or gasoline. My electronical, mechanical, perpetual motion machine. Everyone said that it couldn't be done. It only took me a year with only limited funds. The people at Home Depot all know my name. They didn't know what I was building. They thought I was insane. I care about the air and it keeps it clean. My electronical, mechanical, perpetual motion machine. My electronical, mechanical, perpetual motion machine.
breaks.
Don't talk, don't talk, don't talk. Yeah, nobody can hear me but you. Oh, nobody can see that expression on your face. You want me to continue playing to the end?
Sure. Or how much longer does it have?
I think it's over.
And we're back. This is This Week in Science. If you just tuned in, we talk about science. That's about it. I'm Dr. Kiki, and Justin and Allie are here. We have science stories. How's it going? Justin's just like taking a nap here today.
I have stories. I'll be honest. I'm ill prepared today.
It's okay. You have reasons. You have good reasons.
I have personal reasons and excuses too. I know for my doctor.
Awesome. Let's see. So I have a story that I think is kind of fun. If you want to hear it. You guys know the dark star? Remember the idea of the dark star? I think there's a Grateful Dead song about the dark star. And there's this idea that there's this dark star that we can't see somewhere out around our solar system, and it's affecting the planetary movements, and that it's going to, I don't know, it's probably related to this 2012 Armageddon type thing somehow, I'm sure. Anyhow, astronomers are interested in finding out not necessarily about the dark star per se, but about our solar siblings, which is kind of cool. You kind of think of yourself or our sun and our solar system as part of the Milky Way galaxy. We're on one of the spiral arms. We're just kind of out there doing our own thing, going our own directions. But I think people forget a lot about where our sun may have come from by looking around at other galaxies outside of our own. We've been able to find that that stars are actually formed in this crazy chaotic process where there are gas clouds, the dust, and there's lots of gravitational forces moving and pushing things together. Pressure. And that stars get spit out of these crazy chaotic formational times. And our sun probably came out of one such event, and it was probably shot out of some gas cloud at the same time as a whole bunch of other stars. But where are those other stars? And if we find them, and if we find out about them, can they tell us about our own trajectory and where we've been and where the sun has been? One of the questions about our own sun is that researchers looking at where we are and the relative levels of metals in our solar system, that it seems a little bit aberrant. So is that something are we like other stars in that way, that their solar systems have high levels of metals? Is that something that came from the gas cloud that we were created in? So anyway, researchers published in archive.org, Anthony Brown at Missouri State University and some of his researching colleagues, they have done an analysis of what's called the Hipparcos. I believe it's the Hipparcos catalog. And Hipparcos catalog is based on a space mission, the Hipparcos space mission from the early 1990s, in which it went out and cataloged a whole bunch of stars. Now, there are, I don't know, let's see, researchers are saying that the sun probably had somewhere around 1,000 or so siblings. And then those siblings are going to be spread based on the gas cloud they came out of, probably over about 3,000 light years, all over the place. And the researchers, Anthony says that this space contains 100 million stars. The Hipparcos database contains 10,000. So there's an order of magnitude in terms of star data that we're missing here. And unfortunately, They looked around. They were looked for long lost relatives, looking at their velocity, trajectory, all sorts of factors in these stars in the out of the 100,000 stars that are in the Hipparcos database. And they found only one that was the same age as the sun and had a velocity that was kind of like our own. That would suggest maybe they came from the same place. This is called HIP 21158. It's a white magnitude 7 star in the constellation Taurus. And even though they've found one possible sibling, they say it's probably not an actual sibling because it's moving a little bit too fast to have probably been spit out by the gas cloud at the same time as our sun. So, says Brown, this means that we have not found a single convincing solar sibling within 100 parsecs from the sun. It's kind of sad. We're still orphans. We're all alone. All alone.
Are we orphans, or is it possible that we don't get spit out of a dust cloud together with a bunch of other suns?
Maybe. Where else would we have come from, though?
Well, I mean, just because that's if we're not seeing, siblings of these other stars being spit out at the same time that have trajectories that are where we would expect them to be, or at least within a certain range, and maybe that's not how things started. I don't like the dust cloud forming suns and then spitting them out. Somehow that just doesn't.
I like it. I like it.
Really?
It's really intense birthing process.
In the beginning, there was a dust bunny. This big. And then you were born.
That's right.
Really? That's
you were
how it all
spit out.
started,
You
huh?
were spit out of the chewing cud of a dust bunny.
But he forgot to bring out the sweeper, and next thing you know, they got a solar system. Cool.
That's right. Thank goodness for the prolific birthing of dust bunnies everywhere. Anyway, there's a launch of the new mission called Gaia, set for 2012, which is interesting. And an interesting date for that launch. Anyway, it's going to go out and catalog about 1 billion stars, which is going to give us a much larger database to work with. But the problem is that database is not going to be ready until 2020. So we're going to be out of luck in terms of having more stars to look at to see if if this dust bunny hypothesis actually holds any weight. For 10 more years.
Start calling it the dust bunny hypothesis.
The dust bunny. The great intergalactic dust bunny. It's perfect. Perfect.
Do the scientists have any idea as to why there are no neighbors? Do they have any hypotheses?
Oh, that's a really good question. In terms of why there are no neighbors, in this here, it doesn't say. Yeah, it doesn't say that. Basically, what they're thinking is that we've been around for 5 billion years, and that's probably where the Milky Way has traveled and how the sun has traveled within the Milky Way and where we are. This probably has, covered hundreds of thousands of 5 billion years, maybe.
Mm-hmm.
Travel. So things are probably very widely dispersed. But we just have no idea right now. I think they're just really curious to find out what else is out there. I don't know. I think it's interesting.
Yeah, it is really interesting. But yeah, we are, even in our own area, we are very rural. We don't have very many neighbors. I don't know why our neighborhood isn't considered one of the more desirable neighborhoods. Everything looks good from here. But we have, I mean, there are definitely areas of not just this, definitely of the solar system that are more densely populated with solar systems, and areas of the universe that are more densely populated with galaxies. We're kind of out in both rural BFEs.
Yeah.
So,
I'm kind of out there.
Yeah. Which is nice. I mean, there's an upside to that. I mean, the downside is if we're trying to find other solar systems, other planets, and travel to them, we've got a much, 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 much tougher job of doing it That's the downside. The upside is there's less junk out there flying around that can slam into us and, eliminate all life on the planet. So there's an upside to being rural, which actually might give us a much better chance of having had the opportunity to have intelligent life or life at all to have evolved here. Because if you're in an area that has lots of neighbors and you're used to bumping into each other a lot, it's not too good for most of our evolution. Bumping into other objects has ended badly usually.
Well, except for comets, it might have helped a lot in terms of comets.
This is true. On the other hand, the panspermia, that's all the water in the oceans and our atmosphere came from possibly from comets. So yeah, maybe it's even greater chances of life forming and then getting wiped out. It forms fast, it gets eliminated fast out there in the more urban areas of our universe.
Exactly. Someone, GL Desater in the chat room says 240 million years to orbit the galaxy. The sun's sibling stars would be anywhere in the galaxy after 5 billion years. Yeah, they could be anywhere. So that's going to be an interesting hunt. Hey, Ali, you had an interesting space story about Mars.
I have,
that was kind of cool.
About life on other planets. Basically, Barack Obama has announced that he hopes or he has the intention of sending humans to Mars by the mid-2030s. And so, but first of all, they're going to have to create a good garden on Mars, which will allow life to be there. Basically, a lot of researchers are already testing the conditions, basically testing organisms in really extreme conditions and Mars-like conditions to see what could survive there. And mostly they're testing microorganisms, bacteria. They are finding that eventually they're hoping that a lot of microorganisms can eventually help turn Martian rocks into soil, generate oxygen for us to breathe, purify water, and recycle waste. Basically, just through different microbial colonies of different species that do different things in their metabolic pathways that hopefully they can do this. And there's a lot of different studies going on of the different types of conditions. One is being conducted in the Research Institute of the Open University in Milton Keynes, England. And Karen Olson-Francis is part of this team, and they study, they basically subject terrestrial organisms to extreme stresses. And they found that after launching a bunch of these rocks, they basically made a capsule with a lot of rocks from these sea cliffs, and they launched them into space. And so after 10 days of space exposure, they found one cyanobacterium that was new to science that they hadn't known of before that survived the ride. And so that was just a first step in finding organisms that can possibly be used in space.
That was new to science?
Yeah.
So they didn't know it was on board? Right.
I guess not. I think it was just a ton of, too many to even maybe count, and then they found which ones were still alive.
Gotcha.
I'm guessing, yeah. Basically, and also this particular experiment provides support for the lithopanspermia hypothesis, which is the idea that living cells can be transported through space in meteorites, and they could carry life to other planets that way. And some people think that might be how life on Earth started. And yeah, so basically it showed that these organisms can survive in the vacuum of space and then start to grow again. So that was really interesting. And
I think that's amazing. Just the idea they can exist in the vacuum of space, but then at the same time, they're also withstanding all the solar radiation, the cosmic rays, like everything that's bombarding them from outer space. I mean, they go dormant and live. That's crazy.
That is actually a huge problem on Mars, particularly because they have really high levels of UV radiation. And in this particular experiment, actually, I think they had filtered out most of the UV radiation, so these particular organisms didn't have to deal with that. But so they were saying if they did build gardens on Mars, they would have to be in greenhouses, because most organisms still can't deal with that UV radiation. So that is a big problem on Mars. Um, but they're also looking at photosynthetic cyanobacteria, largely because basically if they go to Mars, they're going to need some kind of primary producer that can produce oxygen from carbon dioxide, and so yeah, they are doing this experiment with a self-contained recycling system designed by ESA called the Melissa Loop, or Micro Ecological Life Support System Alternative, which recycles human waste into water, oxygen, and nutrients through the use of cyanobacteria. Yeah, and they're used to make the protein-rich food supplement spirula, which ESA has named as one of the 9 essential crops to grow on Mars. So they're even planning out which crops they're going to want to grow for the astronauts there. And Mars isn't actually that terrible of a life support system. They have the UV is bad, but they have a lot of CO2. The atmosphere is actually 95% CO2, and the equator at midsummer, the temperature can reach 20 degrees Celsius, and the atmosphere, 20 degrees Celsius. So that's not too bad.
It's kind of balmy.
Yeah. Yeah, and another major problem on Mars is there's no soil that could grow anything. It's pretty much pure rock, basaltic rock. And so researchers also think the solution to this might be bacteria as well, because there are actually bacteria that can turn rock into life-supporting soil. And some studies have been done on this on Earth already, looking at Icelandic basalts for organisms capable of surviving on a diet of what would be Martian, similar to Martian rocks. Yeah, and he found that one researcher, Wilkinson, found that there were some organisms that could grow in this media rich in heavy metals such as copper, nickel, zinc, and chromium. And basically, it suggests that space gardeners could also use the sort of bacteria on the Martian rocks.
I love that. Yes, you have to have bacteria that can do anything. I mean, we're going to have the bacteria that can eat the rocks. You have the rock eaters, and then you have the bacteria that can actually survive in the soil that's highly metallic and be able to actually produce a more suitable soil. And then you have to have the carbon dioxide photosynthesizers that are going to create more oxygen for you, and step by step by step. This little,
all through microbes. It's crazy, yeah.
Yeah.
And on the trip there, we can grow strawberries.
Oh, yeah.
Yeah, NASA's working on maybe it's West Lafayette, Indiana, somewhere. Working on a low maintenance strawberry that could be grown in space.
Astronauts like strawberries. Sure, why not? I mean, I don't know. I wonder if they're going to end up like the big, without gravity to contend with. I wonder if they're going to end up like big strawberries, or if they're going to be like the little tiny strawberries that are nice and sweet. I don't know. Yeah. I wonder what problems there are to growing a strawberry in space.
Haven't they had problems with plants in space before, though, not knowing where up and down is for the seeds?
Yeah, I think
to start growing.
Yeah. They have the
like
roots and the shoots have to figure out. Yeah, go ahead.
Mostly, but then if you get like a little fake fan on there or something, I'm sure they can figure it out.
I don't know. You have to. I think that'll be that'll be the interesting challenge: getting plants to grow in the right direction and getting them to, getting little farms in outer space. But that's what we need. We need a little, herb garden, strawberry patch on the International Space Station. I like it. I like it. And then on to Mars, because if you can make a strawberry in a spaceship, you can get to Mars, and you can put bacteria there.
And lichens, actually, they actually found that lichens are the real kings of rock processing. That lichens are actually the ones that basically eat up the rocks the most effectively, and also that they've been shown that they can actually survive in space. In the first experiment they were used, and then also that they can survive in similar conditions to those on Mars, but they still can't deal with the UV radiation as well. And they were able to photosynthesize as well. So lichens could be a really big asset to the new Martian community as well.
I think it was one of the only plant lives in I remember seeing in Greenland.
Lichens?
Yeah.
Yeah, well, it's so cold, and the amount of daylight that they get throughout the year, that would be limited, and that kind
It'd be 24 hours for like half the year.
Half the year, and then nothing for the other half.
And then complete darkness. Yeah.
Yeah. I wonder with Mars if it would, if we'd have to create more of an atmosphere, use bacteria first, create more of an atmosphere, and then you move up to lichens. Because an atmosphere is going to help protect you from any kind of UV radiation. So create more of an atmosphere. It shields you a little bit more. And then you bring the lichens, and the lichens help a little bit more. The atmosphere grows a little bit more. And then you start bringing, people, cockroaches after lichens.
You're already on Mars. You're not going to be able to go outside for a really long time. Like forever. We should just live underground. Forget the whole surface of the planet. And then as long as we're going to be living underground, we have a perfectly good planet to live underground here. Why don't we just start...
start digging. Just start digging. Become the tunnel people.
That's where we should... That's pretty much what Mars is going to be. Mars is going to be like living in any city in America or anywhere else in the world, but where you just can't go outside. Why be on the surface at all at that point?
And at that point, they'll probably have pretty good artificial outside experiences that you can go through too, like in, I don't know, the form of like a movie sort of, but ultra 3D sort of like. Yeah, so you would be able to have the outside experience.
Allergies and pollens and dust and all that nasty stuff.
Sounds like heaven. Wow. So funny. Hey, Justin, did you have another story? We're getting down to the end of the hour.
Oh, we can do the mailbag if we've got if you've got that ready to go.
I have got the mailbag ready to go. So this is a letter from Mike Sherman. He wrote in, said he'd been working on a project involving tissue damage from touching hot surfaces. So your question from last week, we had a question of why 108 degrees Fahrenheit. That discussion caught his ear. Proteins denature at temperatures of about 43 degrees Celsius and above. 108 degrees Fahrenheit is 42 degrees Celsius. So there is a reason for development of temperature sensitivities at that temperature. You get higher, you get 108 and above, and you're going to be denaturing your proteins. And that's never any good at all. So thanks, Mike, for that. That was a great answer to that question. Hello, my name is Giannis Guerra, longtime listener. Hi, Giannis. I've been a little bit delayed in my TWiS listening, just going to the one where Allie was going to review climate change and was reading an interesting article at the same time. Public debates driven by incomplete scientific data, the cases of evolution theory, global warming, and H1N1 pandemic influenza. Interesting. That was in archive.org. It shows that to adopt a cautious, balanced attitude based on clear but inconclusive data appears to be a lose-out strategy. In contrast, overstating arguments with wrong claims, which cannot be scientifically refuted, appear to be necessary but not sufficient to eventually win a public debate. And he asks, is this why we are having so much trouble convincing people about topics? Should we be more forward with our certainty about the global picture in evolution or in anthropogenic global warming and let the little disagreements fall off of the main discussion? How do we link this to our ethics as a scientist? Interesting topic. Would like to know your opinion and how this could affect how we communicate, how we communicate scientific data. And I think it's a big question. I think I don't know. I think it definitely should influence how we communicate scientific data, and that these kinds of things need to be taken into account when we start talking to people about things that don't have absolutely 100% rock solid results. I don't know.
Yeah, but the problem is you have people who will speak with great confidence, and that has a gravitas to it and a sense of authority and a sense of finality where people will believe that. If you
Yeah.
say something very confidently to somebody, like at nighttime, it doesn't get any darker. It's just in your head. It almost sounds for a second like, could it be? No, okay, it does get darker. But even in then, something as ridiculous as that, if you say it, that's, confidently,
Yeah.
it'll sound true for a second. And I think the
Yeah,
problem with science, and I think he's a great point, is that you're not speaking just for yourself. You didn't like come to your conclusion and say, "Yep, I looked at every piece of data, and I'm now saying that it's all correct and it's all 100% in this direction." Because you did your portion of it, you did your piece. There was a research team over here, there was a researcher over there, there was the whole university worked on it, this portion of it over there, and it's all come together and put into this final thing, and it all adds up to global warming. And so there should be somebody, lots of people out there as sort of lobbyists, spokesmen for the whole group. When you have scientists talking on behalf of other scientists, a lot of times they throw in all of those hedge words. It's so like, it may be leading to an indicator of, and could be, it's got all those disclaimers in there for some reason, which scientists are used to because they're used to getting new data tomorrow that sets them off in a couple other directions, and then they fine tune it so they don't try to lock themselves in too hard in their own thinking. But yeah,
Right.
I think global warming, we should just all get together, pitch in, and find like a front man, somebody without political baggage that could just totally go in there and like that guy from what was that movie? What is the
Without
the
political
cigarette
baggage,
lobbyist?
but with big guns.
No, the celebrity spokesman. Who is the thank you for smoking? Look at that guy.
That's great.
That's a great way. Look at that guy to go out there and just tell people that global warming's happening.
So what you're saying we need to do is hire the cigarette industry to tell the global warming. It's not global warming, it's climate change, the climate change story.
Mm-hmm. Exactly.
Yeah.
It's not even climate change. It's still climate change is too weak. Climate change is an adjustment on your air conditioner. Right?
All right. Moving on to the
Chlamydia.
last.
It's chlamydia.
Chlamydia.
That's
That's
what
right.
the planet has. That's what that's if you don't want chlamydia, then you need to act fast, people.
That's right. Chlamydia is a dangerous disorder.
Wear your
Last
yeah.
mailbag letter says, suspect that I am just emailing the ether here, but just in case. And so this is why I put this in here. This is not the ether. This is the show. Archive is pronounced archive because the X is the ancient Greek letter chi, pronounced chi. It's like when you say a chi or X squared statistical analysis. Regards, Steve, the moon explosion guy. Thanks, Steve. And on next week's show, we're going to have more science infotainment and shout outs. I have a couple of shout outs to Ed Dyer, David Eckerd, Tony Steele, Giannis, and Mike. Thanks so much, everyone, for sending in stories. Sometimes I think we just need more than an hour. I don't know. There's so many stories. I love them all. They're so great.
Yeah, and next week I'll have some stories that I've had a chance to read before going on the air with them.
That can help a little, just a little bit.
Oh, Allie, are you going to come back with us next week?
Yeah, I hope so.
Right
Plan
on.
to.
We've got Whole Earth this coming in, right?
Yes, this is true.
Yeah, I hope we're in condition to do a show after that. It's a fan. If you haven't been, it's a fan. The Whole Earth Festival in Davis is something not to be missed, unless you've never been there, in which case you're not really missing anything. Well, you don't know. You don't know what you're missing if you haven't, experienced
Justin.
it first.
Justin,
Yeah, are we done?
end the show.
Thank you everybody for listening. We hope you enjoyed the show. You can, if you got any suggestions for somebody we should interview, stories we should cover, you can email us at Kirsten@thisweekinscience.com or Justin @thisweekinscience.com.
Yeah, we love your feedback. And we hope you have enjoyed the show. We are also available as a podcast. You can go to our website, www.TWIS.org, and click subscribe to the TWiS Science Podcast. It's that easy. It'll give you information on how to subscribe, or just search for This Week in Science in iTunes. And if there's a topic you'd like to cover, let us know.
Yeah, or forget the emails, though. Do we listen to those emails anymore? Just hit us up on the Twitter at, what is it? What do we got? Jackson Fly and Dr. Kiki.
That's right.
Yeah,
and
those are,
we will
those are
be.
easier.
That's right. And we will be back here next week. And we hope that you will join us once again for more great science news.
And if you've learned anything from today's show, remember,
it's all in your head. We made it through the wilderness.
Great show.
We made it through.
Oh my God. I think I got a lifetime of allergies all in one day.
Oh, that was me yesterday. It was an awful day of allergies. There's a study this last week that it's about immune systems, and it suggests that Justin's totally falling down. That the immune system is actually triggered when you see somebody else who's sick. So if you see somebody who's sneezing or coughing, has a runny nose, that your immune system is going to get more, pumped up and active. And if that's the case, then if people have allergies and they're not really sick, but they look sick because of their allergies, then your immune system is going to become more active, and maybe you'll be more likely to be triggered by an allergen because your immune system is waiting for something to attack. So this is my own little personal idea that I'm coming up with here, that really this surge of allergies and how allergies are growing and getting more and more and more. This is what it's related to.
I'm actually worried I've encountered some sort of ancient evil. I've been digging around. I was digging around in a very old barn today, and because I had some stuff that was boxed up in storage in there, and I think I always wonder, what if there's some kind of just soil, back microbes that just have been hidden for 4 billion years until Justin Jackson went digging around in that one spot, and now I'm gonna have some horrible disease because of it.